Hello and welcome to Roundtable, the most dangerous animal on the planet. That's us. We are destroying habitats, pushing more and more species to the very edge of extinction, and a typical human response? Just make more creatures. Today, we're talking about cloning. The International Union for Conservation of Nature predicts that by the end of this century, nearly all critically endangered animals, those already close to extinction, and almost 70% of those whose decline is being closely watched, could be lost forever. For example, the latest estimates say that there are as few as 115,000 hippos left in the world. Some scientists see cloning as the best way to preserve endangered species, providing the means to bring them back to life. Good to have your company. I'm David Foster. With nearly 42,000 species on the IUCN Red List of endangered creatures, scientists are desperately trying to keep them all alive. Well, a controversial branch of conservation research has led some scientists to advocate cloning endangered, even expired species to reverse the impact of what many now believe is the sixth mass extinction. Those opposed to the idea say that humans should instead stop biodiversity loss by tackling the issues causing it in the first place. How realistic will this approach be? And how can scientists be sure that cloned animals won't die from the same pressures or be able to mimic learned behaviors of the past? Let us talk then about what has gone before and what may come in the future. Let's go to Melbourne, first of all, Australia. And Andrew Pask joins us from there, Professor of Biosciences at the University of Melbourne. He's trying to develop technologies to bring back Tasmanian tigers from extinction. In London, Dr. Kalpana Surendranath, creator and leader of the Genome Engineering Laboratory at the University of Westminster. And then we head out to Estonia, to Tallinn, and say hi to Tiit Maran, director of Tallinn Zoological Gardens, a European mink biologist who thinks, I believe, that we should do more to protect those that are still alive than to bring back from the dead those that have gone before. Andrew, I want to come to you first of all, and before we talk about the Tasmanian tiger, let me read out a little list that we prepared of endangered animals. The Amur leopard, Russia and China, that's where that is. 2014-2015 thought that less than 192 were in their natural areas. Now 84, so the number's still going down there. The Javan rhino, and you find rhinos as well as those ones in Africa and Asia, but the rhino in Java closest to extinction, 46 to 66 left. The orangutan in Borneo and Sumatra, 230,000 in total, their numbers, around about 105,000, that is the Borneo one, the Sumatran one down to 14,000. And how about this? This is a creature I didn't know about, the Cross River gorilla, Nigeria and Cameroon, round about 200 to 300 left in the wild. Now, the Tasmanian tiger, I said it is extinct. Some people believe it could still be roaming the wilderness parts of Tasmania, but last seen, I think, in 1930. How are you going to try and bring it back, Andrew?
Yeah, I mean, I think all those animals you just mentioned are great examples of how we're on this brink of this major loss of biodiversity on our planet. And unless, as scientists, we're able to step in and help manage and really help these animals, we're going to lose even more. So what my lab's really interested in is trying to bring animals back that we tragically have lost. And a great example of that is the Tasmanian tiger. It was, lost in 1936, human-driven extinction. We still have heaps of specimens that are very well preserved in museum collections. And so we have the ability to apply modern day technologies to try and bring some of these species back that played really critical roles in the ecosystem. They're really important for conservation.
Are you doing
in their
that?
Own right.
Are you doing that now? Are you taking the DNA from these long dead creatures and using it in laboratories. You're nodding, so you are doing it. And how successful is it?
Yeah, so we've sequenced the entire genome, so that entire blueprint of how to build a thylacine, and we were able to get that from these specimens. And we are now looking at potential surrogate animals that can serve as a surrogate genome and perhaps as a surrogate mother to actually then start the technologies of trying to bring these species back.
And what would happen then that those animals that came out would be clones of the original animals, but then carried by different creatures? Would they be able to breed and therefore extend the population even further of the Tasmanian tiger, also known as the Tasmanian wolf, by the way, isn't it?
Yeah. And I mean, that's the whole idea. The ultimate goal of these projects is not to just bring the animal back, but to actually get to that point of having an animal that you can put back into the population again, or into that ecosystem, going to that next step of rewilding these animals. But it is different from cloning. They are not exact clones, and that's one of the reasons why we don't want to have any more extinctions, because it's never going to be as good as that original animal. You're essentially have to engineer these animals from the ground up. So they're, enormous projects, and only ones that you would want to consider for a species that was so critically important in that genome.
Andrew was talking there about the way that he's hoping to bring back to life Kalpana, the Tasmanian tiger. I thought it was cloning. He tells me it's not, but I would think a lot of people are as confused as I am. What is the difference between what we've just heard and what I described as cloning at the very beginning?
Yeah, I mean, there are different types of cloning. The term cloning refers to many different things as a molecular biologist to me. But in this context, when Dolly was created, let's say...
Dolly the Sheep back in 1996. We should point out to those with not very long memories. I can remember it because we were so surprised at it. But that is Dolly the Sheep, the first cloned animal.
That's right, yeah. So when the first cloned animal, Dolly, was created, scientists just used the DNA, the entire DNA content of a somatic cell, and created a replica of it through cloning. But in this case, what we are looking at is introducing desired properties into the existing genome of an extinct species. So this is different from just cloning an animal, which will create an identical twin, if you like. Whereas engineering an organism will lead to new features, new characteristics.
So this is gene editing, where you can take the very best of what you want and leave out the things that you don't want. It's called CRISPR technology, isn't it? CRISPR. So that's the cutting edge. Is it being used a lot at the moment, Kalpana? Because I see you mentioned that there are dogs, there are sheep, there are, well, countless animals where it's being done. But the question is why?
No, the classic cloning experiments have been done in many of these animals, whereas using CRISPR for engineering animals is limited. So we have examples of farmed animals which have been created using CRISPR, particularly to improve farming practices and stuff. Yeah, we do have a lot of examples, chicken, many farmed animals, yeah.
And that is because they would lay better eggs, I assume, not because chickens are likely to go extinct fairly soon. But this creature, we're going to see from this chap who's just opened a museum in the United Kingdom with thousands of fossils in it. His name's James Hogg, and he's got a 180-million-year-old Ichthyosaur fossil that he discovered was actually pregnant when it died. He has hopes for the future for this old beast.
Hello everyone, and welcome to the Yorkshire Natural History Museum. Now we've got something very special to show you today that has recently entered the news, and that is this specimen right here. This is the inside remains of an Ichthyosaur, a creature like this specimen behind me which we have the skull of, and similar to this model right here, an extinct marine reptile. What's special about this one is it's got the preserved embryo inside it of a young juvenile that would have been birthed into the world had the parent not sadly died. Thank you for looking, and if I can say one thing, wouldn't it be incredible to have these animals alive once again today?
He thinks, and he's been told that it may well be possible, to breed another version of this marine reptile. with what he's got. Do you think that's likely?
Yes and a no. It depends on how intact the DNA is. If they are claiming that it is closer to reality, it means that they have a good intact DNA, because dinosaurs lived far away from us in terms of the timeline. So it would be interesting to see how intact the DNA is, in the species.
I have to say, in researching this, I've discovered it isn't actually a dinosaur. It's an aquatic reptile. I'm sure there's a small difference for those that study these things closely, but the point is, it's actually a fossil. So it's in a piece of rock, or it is a piece of rock. Is it likely that the DNA would still be there?
Well, DNA is made of chemical substances, right? They are perishable. So they are susceptible to external forces. So it will be interesting to see. They are actually perishable materials because they are chemicals, adenine, guanine, thymine, all these small molecules that make the DNA. They are basically chemicals.
Tallinn Zoo, you're looking after the European mink. First of all, tell me how dangerous it is and why you think that it's better to keep alive those species that we already have rather than try and recreate the past.
Well, I've been working on conservation for 40 years, and mostly was working for one species, European mink, which is one of the most critically endangered small carnivore in the world. And, if you're looking on the pace of biodiversity decline, and at the same time, we would like to start to implement fancy technological approaches. The thing is that everything is disappearing so quick that we are not able to use very expensive technology everywhere. And also, the technology usually, if we talk about species conservation, we are talking mostly about the species like some evolutionary unit or a taxonomic unit. But the species make sense only with relations in ecosystems. And if we want to resurrect some of the species which have been decided, we have to ask from ourselves, do we have ecosystems which could actually support these species?
This is the danger, isn't it? You bring something back to life, or you bring back many, many more of them, and there's nowhere for them to live, and there's no proper way for them to interact with other species around them that may have become more dominant.
Well, we don't know, but the basic thing is that we have to think in terms of ecosystems. If the species is gone, there could be different kind of reasons. There might be, for me, certain very few points when we could actually use cloning to bring back something, maybe. But at the same time, like the mammoths, where we put the mammoths when we bring them back? We don't have ecosystems like it was in the earlier times. So we also always have to think what we are doing. And the second part, which I'm kind of critical about this kind of high-tech approach, is that it will, by what you call the possibility to say that it's not a problem. Even if the species goes extinct, we could bring them back, which is actually not right. And that will give kind of wrong understanding about the biodiversity crisis in which we are right now.
Andrew, I want to ask you about your particular continent. We're going to hear from Australia's Environment Minister first, and then your thoughts.
We deserve to know that Australia has lost more mammal species to extinction than any other continent. We deserve to know that threatened communities have grown by 20% in the last 5 years, with places literally burned into endangerment by catastrophic fires. And that's why I'm also happy to announce today that we will expand Australia's national estate. Our government will set a goal of protecting 30% of our land and 30% of our oceans by 2030. We'll explore the creation of new national parks and marine protected areas, including by pursuing the East Antarctic Marine Park.
So there you have the Minister saying, listen, we are doing something, but is it enough?
Yeah, I think we're changing the world so quickly these days that the species can't evolve and adapt to keep up. And that's really part of the huge problem here. So one of the things we're doing in Australia in particular is we know because of raising global temperatures, we're going to have more adverse weather events, more bushfires, more potential extinction events as a result of those. So we're going out and we're collecting tissue from those animals in vulnerable areas in the hopes that we'll be able to use that to bring those species back one day. But we still lack those technologies of taking a cell from a marsupial species and turning it back into a whole living animal. But that's exactly the technology that we're developing and that which we need to one day de-extinct a thylacine. And so one of the things I like about these projects is they have, or they're developing technologies that have immediate conservation benefits right now, as well as one day underpinning these de-extinction events. But I totally agree. The best thing we can be doing for biodiversity is not to lose any species. But where we have lost some species, and more recently than a dinosaur, for example, we actually do have the opportunity to bring some of those back. And for an animal such as the thylacine that I'm working on, that ecosystem is still intact, where you could put that animal back into that environment. That food chain is still there. And we do have the opportunity to hopefully preserve that ecosystem even further.
But just remind me, you said at the very beginning why the Tasmanian tiger, the Tasmanian wolf had disappeared, and it was because of human encroachment upon its land. Therefore...
No, they were aggressively hunted to
Okay, they were
extinction,
aggressive. So therefore, how do you stop that happening again unless you bring in new laws and et cetera, et cetera, et cetera?
Yeah. So obviously we would protect if we were to return I think we now appreciate just how incredibly unique and important that animal was. Whereas, back at the late 1800s, early 1900s, we just didn't have that appreciation of how important those animals are in an ecosystem.
And they're not dangerous?
No, not at all. They're like a medium-sized dog.
Okay, so's a dingo.
Yeah.
Kalpana, let me come to you and talk to you about whether, in fact, we're doing this for our own benefit rather than for the benefit of the species as a whole. I've got this fact here. 2 beagle dogs, 2018, you mentioned beagles earlier, South Korea, are being created from cloned skin cells altered by gene editing, your speciality. And scientists hope, this is what I'm told, that the technique could be used to eliminate disease-causing mutations from pedigree dog breeds. That's taking it too far, isn't it? That's being done simply for cosmetics, for the owner's benefit.
That's right. But I can't dismiss the fact that gene editing is used for cosmetic purposes. We have glowfishes. We have all sorts of examples for cosmetic purposes. But should we invest the resources and time in such research is a question. Yeah.
Does the sort of work that you do benefit humankind in a sense? Because earlier on this year we had a man who was given a pig's heart. He didn't last very long. This was a heart transplant. He didn't last very long, but they were very hopeful. And now I read that German scientists are planning to clone and breed pigs simply for their hearts so they can be used, and they'll be able to perfect this, therefore making human organ transplant that much easier.
Yeah. My research doesn't involve whole animals. However, I can very proudly say that I'm very close to contributing to the betterment of human welfare by the recent discovery of new drug targets for colorectal cancers, one of the 3 top cancers in the UK. So laboratory, in a very controllable way, depleted certain driver genes, and we have demonstrated that these genes can be targeted specifically for curing these type of cancers. So, of course, yes, my research, I proudly can say that, is closer to serving the society.
And that has happened. That has happened by extension of the fact that more research is allowed to be done on animals than on humans. Is that fair?
I would say so, because because I work with the cultured cell lines. The next step would be to definitely demonstrate this in animal models. And we will be collaborating with experts and demonstrating this in animal models, yes.
Extraordinary. So we're talking there about saving humans. Tiit, I want to come on to the work you do at Tallinn Zoo. You say the nature of zoos have changed over the course of the last 50 years enormously. They're no longer just places where people come to ogle, or where zoos say, just for cosmetic purposes, to go back to where we were, that actually we're trying to save the species, but where in fact they're just trying to sell the experience of people looking at them. What are you able to do to help protect?
Well, so what they can do is basically two things. One is to try to keep animals in captivity, and not animals, but populations, what is important to emphasize, in case it's possible to bring them back into the wild. But what is even more important is actually to educate people about the biosphere, about the ecosystems, and the need to maintain biodiversity. And it's interesting, in the 70s, when this kind of new paradigm actually came to the zoo, there were always this kind of strange announcement that the conservation is very easy. Let's bring all the animals into the zoo and the conservation problems are solved, which of course is not the case. And it took some while to understand that what we can do in a zoo is actually very small things. Bringing animals back into the wild is basically like a fire brigade work. We can do something, but in long term, to maintain biodiversity, it's not the help of technology, either of captive breeding or cloning. It's basically the way how our societies are functioning, which basically comes to the level of the mindset, the culture, what we have, and how we relate to the surrounding.
Yeah, to take your analogy of the fire brigade, it's to advise people how not to start fires in the first place, rather than to have experts to put them out.
Yeah, basically it is to say that we can do a lot of fire brigade work, but it doesn't help much if we don't teach for the people how not to play with matches. We will have more and more problems, and nowadays, if you look around, we have more and more endangered species problems and way more complicated to solve them. So again, we could make, this kind of focused approaches, but in long term, everything depends how the society is actually behaving.
Okay. Quick way for you two, that's you, Andrew, and you, Kalpana, to bring this to an end. I'm going to ask you, Andrew, about the tiger. That's a Tasmanian tiger clock behind you. I know it's showing the wrong time, but as far as you're concerned, any time is good time when it comes to Tasmanian tiger. When might we see one?
I would hope that this technology is advanced enough that sometime in the next decade, we would start to be seeing animals de-extincted. Whether the thylacine is going to be the first one or some other species, there are many labs working on this technology. And it will just be another one of those tools in our conservation toolkit that we can apply, as we talked about today, in specific circumstances where that animal was really critical for that ecosystem.
Kalpana, you know where I'm going with this, because I'm sure you've been asked it many times. Once you've cloned the Tasmanian tiger, once they've cloned Dolly the sheep, once they've done other animals as well, chickens, whatever it happens to be, the conversation turns to humans. There will be people out there, unethical. I know there's an awful lot of legal structures at the moment to prevent this from happening, who will think now's the time to do it. Do you think that will happen?
Maybe in a few hundred years. I can't predict it at the moment. I always insist on one fact, that neither the genome nor the CRISPR tool have been characterized fully. We don't know the hidden secrets of the genome. And also, we don't have full control of this genetic scalpel called the CRISPR tool. So once we have that in our hands, probably it might be a sensible thing to do. Even in that case, why should we want to do it? What do we want to achieve by doing this? There's a lot of ethical questions around it. So I would still think that it's an open question. We'll have to wait and watch.
You say you believe in science, but you trust in nature. Therefore, if you were gene editing humans to make them better and less susceptible to disease, that would be believing in science rather than trusting in nature. It goes against what you actually really want to see happen, doesn't it?
If it can stop needless human suffering in a tightly controllable way, I mean, with all the guidelines and restrictions in place, no gray areas whatsoever, but we might take a very long time to reach that stage.
Tiit, let's go back to the animals. I know you think you would rather concentrate on the conservation rather than going back and sort of manipulating them scientifically. But if it helps save species rather than bringing them back to life, would that be a good thing?
It's a difficult question. And I'm just wondering, the ideas of what we have today, can we be sure that we can say that these kind of things we are doing nowadays, or we plan to do, can be regarded to be the best way to handle different kinds of life forms? If you come back to the conservation issues and use technology, especially the gene editing, it's starting to be a very complicated issue because it's kind of playing a god, like a mastery over the nature. And for me, it seems to be extremely dangerous to do it.
Okay, there you go. Thank you very much indeed. The moral as well as the practical questions being asked and answered here on Roundtable. Thank you so much from Australia, from London, from Estonia. We thank you very much indeed wherever you're watching this edition of Roundtable. Until next time, well, I hope there's a next time for you and for me. Goodbye.